Alrighty. Can I have your attention, please? Hello and welcome to our genetics revision lecture for our first semester of medicine. There's a few things we need to cover, but just to give you an idea of how far you've come genetics-wise, you guys have all covered this, or at least I know we did last year in lectures and tutorials and syndicates and whatnot. And there's about 15 to 20 hours of content here. I can't do 15 to 20 hours of content right now. I've got 1 hour. So what we're going to do is cover these 5 main things. And it's your responsibility. What I need you guys to go home and do is fill in all the gaps that I'm not covering today. Go take a look at the lecture slides. Go take a look at the notes that you took in the tutorials and fill in those gaps so you're prepared. All right? So, square one. What we need to talk about first is the central dogma. All this means is inside every cell in your body, you've got some DNA. That DNA gets transcribed into RNA, and that RNA gets translated into a protein, and these proteins are pivotal to your survival, right? But you need to know it in a bit more detail than that, so let's start with this. If you don't already know this, you're in for a world of pain over the course of the next hour. All right, what we're looking at here is our nucleotide, okay? It's our monomer for DNA. It's very simple. You've got a phosphate group, pentose sugar, which is deoxyribose sugar in DNA, or ribose sugar in RNA. And then you've got a nitrogenous base attached to that. Now, why do we care? We care because when a few of these guys hang out together, they have a tendency to create a DNA molecule, a helix, a double helix. And this helix is made by hydrogen bonds occurring between the nitrogenous bases and a phosphodiester bond being created between the phosphate head and the ribose sugar or deoxyribose sugar. Now, something to consider. The hydrogen bonds are far easier to break than the phosphodiester bonds. So when you apply heat to these things, the first thing that happens is they get denatured into 2 separate strands. If you want to actually break them down all the way into separate nucleotide monomers, you need to apply a vast amount of heat. So much heat that it's rarely worth it. Or you can use a specific enzyme. All right, so in each cell in your body, you've got 2 meters of this stuff. There's no way you're going to be able to store 2 meters of this stuff unless there's a very effective way. Oh, the lads have shown up. How good is this? Okay. So where was I? Okay, right. So you need a very efficient storing mechanism if you're going to be able to get 2 meters of this stuff into all of the cells in your body. How that's done is it's wrapped around these histone proteins to create something called the nucleosome. And this complex of all these histone proteins and DNA creates something called chromatin, and that is packed into this X-shaped thing that you see here that's called the chromosome. Now, it's only X-shaped in the lead up to mitosis or meiosis. Otherwise, it's actually just a single rod. And they're all packed into the nucleus, as it says there. Now, when you need to replicate this DNA in the lead up to mitosis or meiosis, all of that should be happening in the S phase, the synthesis phase. Now, we're not really going to be covering mitosis and meiosis in an incredible amount of detail because that was like knowledge that you've already done. You either did biology unit 3, 4 in year 11 or 12, or you did the bridging course, because we did do that. Yeah. And I'm going to cover it in a very rudimentary way, but if you feel like you don't understand those 2 things, you need to go back, cover the notes that you've made, or take a look at the bridging course so you understand, because all the concepts we're talking about today are building on that knowledge. I'm not going to be focusing on mitosis, meiosis so much. But just so we have a good understanding of what's going on in a very rudimentary sense, mitosis is the creation of a diploid cell from another diploid cell. You start with one cell that has 46 chromosomes, and you end up with 2 cells that have 46 chromosomes. And something to consider, this interphase thing that's going on here is not actually happening inside this little mitosis segment. The interphase thing is happening all the way around here, especially in this S phase. So by the time you hit this point here, you're ready to start making the genetic material more concentrated and actually start dividing the cell. Interphase is not happening in this tiny little section here. It's happening everywhere else, okay? And then you've got meiosis. Now, meiosis is the creation of the germline. So what you're going to start out with is some sort of diploid cell, and the goal is to end up with 4 haploid cells. And there's a lot of steps to this, and I hope that you guys have a pretty good understanding of it already. But 2 things to consider. We want to maximize the genetic diversity in every population so that they can survive a long time and survive diseases and whatnot, right? How we do that is during meiosis, you have crossing over during prophase 1, which means that when these chromosomes, right, they're called chromosomes, when they meet up in their homologous pairs, a little part of each of the chromosomes actually swaps over, so you don't have the exact same chromosomes from your parents being transmitted into future generations.
Just 2 seconds.
Yep.
I just need to fix me recording.
Oh wait, hasn't this been live?
This has been, the audio has been live.
Oh, I'm so sorry.
Wait, Karan, did you set up the... Wait, what do you... No, it's not his fault, guys, not his fault. What do you do so far? So you've already set up the... Yep. Yeah, we don't need a camera. Good, good. Let me just...
We need to reassess that guy's important. I think I'm doing really.
It should be now.
You're doing well. You're doing well. It's 6 minutes. I've spent 2 minutes on the slide. Each slide, and there's 7 minutes slides.
Nah, we'll be good. It should be fine.
We
Go
continue.
Go ahead and do it.
All right. Okay. All right. So we're back in business. Okay. So sorry about the technical difficulties. The other thing that happens is independent assortment, which means that you're not creating germline cells. You're not creating these haploid cells. You're not creating one with a complete set of chromosomes from your mother and another with the complete set of chromosomes from your father. There's independent assortment, and the assortment and mixing up of these homologous pairs is random, and that creates genetic variation. Okay, so DNA replication. This is what's happening during interphase, during that S phase, right? So we've got a semi-conservative model, which means that when you replicate DNA during interphase so that you can replicate the entire cell, you're going to end up with 2 strands of DNA, but one strand from each of those double helices is actually from the parent strand of DNA, as you can see in this diagram here. So what happens? The first thing that needs to happen is we need to actually unwind this double helix. It'll become clear on the next slide when there's actually a picture of this going on. So DNA helicase comes in and unwinds the double helices by breaking all the hydrogen bonds between those little nitrogenous bases. And the next thing that happens is RNA primase comes along and sticks an RNA primer where you want the DNA replication to begin. And DNA polymerase then joins the party and begins to catalyze the addition of new nucleotides onto the existing template strand, right? So, and remember, the DNA polymerase can only work on a 5' to 3' direction. It can't work the other way. Why that's important is when you've got your DNA replication occurring, you've got one leading strand where everything's fine and rosy because you're just heading in the 5' to 3' direction. You've also got another strand that's heading in the opposite direction because remember, DNA is anti-parallel. So what you're going to end up with is you're going to end up with DNA polymerase just working on these smaller chunks and creating these fragments. They're called Okazaki fragments. That's not particularly important. I just like saying that word. And then you need another enzyme to come along called ligase to actually bond these Okazaki fragments together so that you don't have all sorts of breaks inside your newly created DNA molecule. What else is there to talk about here? Oh yeah, you've also got a special type of RNA that comes along to deliver these nucleotides as you are doing your DNA replication. We'll talk more about that in a second. And in eukaryotes, like you and I, with a lot of genetic information, during interphase, you need to do a giant amount of DNA replication in the lead up to mitosis. So you're going to have it happening at several different sites, which actually increases the risk of some sort of mutation occurring. And here we go, RNA. So RNA is, think of it like this: DNA is the blueprint to all the, every single protein that you're capable of making. But ultimately, that's not helpful when it comes time to actually make a protein. You need something a bit smaller, something that's easy to transport around the cell. That's where RNA comes into the picture. RNA is a shorter, smaller molecule that also, for some reason, has uracil instead of thymine as one of the nitrogenous bases. And that's what messenger RNA is. That's the RNA that actually provides the information to the ribosome so that translation can occur. And then you've got ribosomal RNA, which just makes up the ribosome, of course. And transfer RNA is the one I was referring to before. This is the RNA that actually delivers new nucleotides when you need them to do DNA replication. All right, so RNA synthesis. This is the first phase of creating a protein. So what you've got is you've got your template strand, and then RNA polymerase actually will unwind and bind a promoter sequence to this template strand. Now, strangely, RNA polymerase works in a 3' to 5' direction, the opposite direction to which DNA polymerase works. And it just adds ribose nucleotides, because remembering that you've got a ribose sugar in RNA, hence RNA, not a deoxyribose sugar. So you get ribose nucleotides added to create this length of genetic material that's called pre-messenger RNA. Now, pre-messenger RNA can't actually produce a useful protein. It needs to be modified in some way. The first couple of things that happen are you get a methyl G cap added to the 5' end and a poly A tail added to the 3' end. The purpose of these is just to make it easier for the RNA to be transferred around the cell to wherever it needs to go, whether it needs to move to the rough endoplasmic reticulum or whether it needs to move to an independent ribosome. Now, the other thing that needs to happen is this RNA, this pre-messenger RNA, needs to be spliced. It needs to be cut up so only the important segments remain and only the important segments are translated. So I was always confused about this. I always thought that the introns are kept in and the exons exit. That's some bullshit. What actually happens is the exons are expressed. That's all you need to remember. Exons are expressed, okay? All right, so then we've got our RNA finally. It's finally ready, it's primed, it's ready to go for translation. What happens here is that messenger RNA shows up at the ribosome, and the ribosome is able to decode it 3 nucleotides at a time. Those 3 nucleotide groups are called codons, and each codon stands for some sort of amino acid. Now, there's a start codon and a stop codon, but that means that there's still 60 remaining codons that can code for an amino acid, but there's only 19 amino acids. So what does this mean? This means that there's a sort of redundancy within this whole genetic system, which means you're going to have several different codons that actually code for the same amino acid, right? So that there's no ambiguity. There aren't codons that might code for one amino acid or another. There's just some sort of redundancy, which means you can have several codons that code for the same amino acid. Right, so if you were to have every single gene in your DNA expressed constantly, then you would have some sort of all the requirements of that and the nutrients for that. You'd be dead within a few minutes. So that needs to be controlled somehow. There's 4 ways of controlling it. So you've got transcriptional control, which just sees transcription factors actually limit the amount of transcription that's occurring at the genetic material level. The other way you can do it is have post-transcriptional control, which just means that you're splicing up the post-translation modification of RNA is just done in a different way so that you have different exons being expressed. And the 2 other ways you can do it is translational control, which just means repressor proteins will bind to mRNA so that it can't show up at a ribosome and be translated. And the final way is post-translational control, which just means proteins have already been produced and we realize, hey, we don't need all these proteins. So what we do is we get protease to break down those already made proteins so you don't get a buildup of useless proteins. Alrighty then. So the next thing we need to consider is patterns of inheritance. And the idea here is just, there are several different concepts here. The first of which is Mendelian Inheritance, which is a very simple recessive and dominant traits. And the other thing, couple of things to consider are polymorphic traits, which are a bit more continuous. They're not binary. There's not it's not like you either have diabetes or you don't have diabetes. It's more like height or something like IQ. We'll get into that in a second. The first thing you need to understand if we're going to be studying inheritance is genetic terms. Now, I'm not going to run through all of these, but a couple of important ones to understand for now are the phenotype, which is just the physical result of the genotype, and the genotype is just the genetic composition. And the other thing to consider is alleles, which are just alternative versions of the same gene. And we'll get into homozygote and heterozygote as we go forward. So in your basic Mendelian inheritance, you've got two types of traits, or you've got two types of alleles. One of them is recessive and the other is dominant. The recessive one is the one that for some reason can, appear in a generation and then disappear for a couple of generations and then show up again. That is because this trait is only expressed in the homozygote. And then you've got the dominant trait, which needs to be expressed in every generation. As soon as a dominant trait dies out within a family pedigree, it's never coming back. RIP. So, and that's expressed in the heterozygous state. All right. So, Mendelian inheritance. So there was this guy that lived in a church. He had nothing better to do than grow peas back in the day. And the situation here is all he discovered was that you don't have some sort of continuous mixing of phenotypes as you breed different sorts of parent peas together. What you actually get is you get recessive or dominant traits being expressed that are representative of one or the other parents. That's effectively what he found out. And he was focusing on autosomal inheritance, which means that these are traits and genes that are based around the first 44 chromosomes, not the sex chromosomes, right? So that becomes important in a second. We'll talk about X-linked inheritance in a moment, but autosomal dominant conditions are generally rare. We talked about they manifest in the heterozygote, and they're equal representations of autosomal dominant and recessive traits within both men and women. There's no gender skewing. And of course, autosomal dominant won't skip generations where recessive can. Recessive can skip generations because you can have a carrier state in a heterozygote. So the heterozygote will have the dominant trait in one of their chromosomes from one of their parents, and they'll have the recessive trait in the other set of chromosomes, and they're going to be a carrier. They're not going to express this trait, but they're going to be able to transmit the trait to their future generations as long as they also mate with another heterozygote who's also a carrier. Now, something that confused Mendel and people that study genetics in general is this thing called pseudodominance, which means that when you get a homozygote and a carrier and you breed them together, you're actually going to get mighty confused about what the dominant and the recessive traits are. So it's important that you use your Punnett squares to figure this stuff out so you're not sucked into this mistake. If it's a 50-50 split of recessive and dominant trait being expressed, you're likely to have a situation where you've got a unaffected carrier and a homozygous creature breeding with each other, okay? All right. All righty then. Moving on. This is what we were talking about before, sex-linked inheritance. So the final two chromosomes in every cell in your body, in the nucleus, are sex chromosomes. So fellas tend to have an X and a Y chromosome, whereas women tend to have two X chromosomes. It's true. It's not funny. It's facts. All right. And why is this important? So if guys only are stuck with one X chromosome, they've only got one opportunity to get these to get the genes on this chromosome correct. They don't have another backup gene lying around that they can rely on. So that is why conditions that are due to mutations on the X chromosome are more likely to manifest in the male. And here, what we're looking at here is we're looking at some sort of pedigree of some royal family. Who cares? But the carriers are all the pink dots. And notice that all the carriers are female. So the female will have this trait in one of their X chromosomes, but they've got a spare X chromosome lying around, so they don't actually express it in the phenotype. Alrighty, but it does tend to manifest in the males, as you can see. Okay, so if we accept the premise that all females have two X chromosomes that have the same genes repeated twice, then why don't they have double the amount of proteins that are coded for on these chromosomes floating around? Well, they need to undergo X inactivation to make sure that their transcription and translation is happening as effectively as possible, which means that you can't have both of these chromosomes expressed constantly. So what you're going to have is you're going to have inactivation of one of the X chromosomes in the female cell, and this tends to be random, which means that if a female is carrying some sort of trait on the X chromosome, they're going to have mosaicism, sorry. Is that how I pronounce that? Yeah, close enough. When they actually express the condition. Now, we'll explain mosaicism in a moment when we actually talk about Down syndrome and related things. And then you've got mitochondrial DNA. Now, mitochondrial DNA is passed down only through the maternal line. So everybody gets their mitochondrial DNA from their mother, and their mother got it from the grandmother, et cetera, et cetera, et cetera. And there are a couple of conditions that are due to a mitochondrial DNA, such as LHON, but that's not particularly important. It's just an example. And now we get stuck into the more complicated stuff, polygenetic inheritance. So not all traits are binary, like diabetes. Some of them are a bit more complicated. Some of them happen on a continuum because they actually have several genes that code for the trait. So this is called multifactorial inheritance because you can have multiple genes that might be affected, that might be leading an individual to have a condition, but then they've got several other genes that seem to be healthy, so they only get the condition sort of semi-expressed, or they only get the trait sort of semi-expressed. And this is what we talk about when we're talking about the continuum. So the you've got sort of a bell curve here where there's no discrete phenotype. You don't have a phenotype that's either one way or another. You've got multiple phenotypes occurring because you've got multiple different genes that are coding for the trait. So, for example, like IQ or height. The other and when we're looking at the classic recessive dominant sort of traits, they tend to be discontinuous. So that's when you've either got diabetes or you don't. So you've got a very discrete definition of two phenotypes. All right, so multifactorial diseases. These are conditions like the neuropathy that we looked at before, but you have an increased risk if you have a family member that's affected, and you have an increased risk if you have multiple family members that are affected. And it may be more frequent in one type of gender, but it's not because it's X-linked. It's just because there's some sort of abnormality that we don't entirely understand as such. And there is a recurrence risk. And if you want to calculate the recurrence risk within first-degree relatives of somebody that has a condition that is of a multifactorial nature, what you need to do is just square root the incidence of disease in the wider population. Alrighty then, so heritability. This is discussing whether a trait is coded for genetically or whether it results from the environment in which an individual finds themselves in. So we're looking at like bipolar tends to have a 90% heritability, whereas asthma only 65%. Now, how the hell did people figure this out? Well, they did their homework. They did their twin studies. There are two types of twins, monozygotic twins, which are identical twins, which means that the zygote actually split apart at some point during the cell division phase, and you end up with two individuals that have the exact same genetic makeup. They're identical twins, so to speak, versus dizygotic twins, which is just a case where you had two eggs were released in the female, and then two sperm came along somehow from the male, and fertilized both of the eggs, and they have the same similarity as normal siblings do. There's nothing more similar about dizygotic twins than just a normal brother and sister that were born at separate times. So what that means is if we follow monozygotic twins and dizygotic twins and we follow their phenotypes and what conditions they do and don't have and what they share and don't share, we're going to have a very good understanding of whether it is a genetic predisposition or whether it's an environmental predisposition that defines whether an individual has a condition. because twins are hopefully brought up in the same environment. Hold on. All right, we're back in business. Now, so when you take a look at your twin studies, you're going to be checking the concordance rate, and the concordance rate is just what percentage of twins have the condition expressed in both of the twins rather than just one of the twins. And if the concordance rate is higher in the monozygotic twins and lower in the dizygotic twins, what that means is there is a genetic predisposition for the condition. If the percentages turn out to be the same, then it means there's actually an environmental predisposition to the condition. So autism has quite a high genetic predisposition, unlike a coronary artery disease, which seems to have both genetic and environmental factors that account for it. All right, population genetics. So what we're talking about here is there's a couple of different things. The first of which is the whole concept of a gene pool, right? So a gene pool is just every single gene that's floating around in everybody's nuclei in the entire population. And what does that result in? Well, you're going to end up with two different things. A phenotype frequency, which is the proportions of individuals that actually physically express traits, and a genotype frequency, which is the proportion of individuals that actually have the genetic makeup to express said traits. And if you want to calculate the gene frequency, all you need to do is get the individuals with a particular genotype divided by the total population. Too easy. All right, now allele frequency is the proportion of alleles in a population, which just means that we're talking about the proportions of the different alternatives of the same gene within a population. So how do we figure out an allele frequency? Well, the first thing we've got to do is we've got to figure out how many homozygotes there are. Well, if we want to figure out the allele frequency for the dominant allele, what we need to do is we figure out how many homozygotes there are and how many heterozygotes there are. We multiply the amount of homozygotes by 2 because they have 2 of the same allele inside their nuclei. But the heterozygotes, we don't need to multiply by 2 because they've only got 1 of each. And we divide that by double the population. And we just do the reverse with homozygotes that actually express the recessive trait when we want to figure out the recessive allele's frequency. Okay. Now, the Hardy-Weinberg law. This law states that allele frequencies will not change in a population as generations go forward, provided some very specific, very abnormal conditions. So the Hardy-Weinberg rarely applies in nature, but it applies in this lecture. So the conditions we need is we can't have any migration because that's going to skew the allele frequencies. We can't have any mutation because that's going to create new alleles. We can't have any selection because that's going to kill off some alleles while preferring others. So as long as we get random mating, no selection, no mutation, no migration, and an infinite population size, this rule applies. Now, in the real world, what does this mean? This means that this is the reason why the dominant trait doesn't eventually just like take over the population, and you don't just end up with like one trait being expressed, even though the dominant trait is expressed in the heterozygote. Well, you're not actually going to end up with the heterozygote just taking over the entire population. This is why, because of the Hardy-Weinberg law. But in a real population, in a real situation, you are going to have selection pressures, which is going to cause evolution, and you are going to have mutations. But the Hardy-Weinberg law is still useful when you're trying to calculate genotype frequencies after you get some sort of migration of a new population out of an existing population. Let me explain what that means. Well, look, I'll explain what it means in a sec. First, let's cover this. Okay, so you've got genetic inertia. This is why the Hardy-Weinberg law is the way it is, because allele frequencies just don't want to change from one generation to the next. If you want to calculate genotype frequencies using the Hardy-Weinberg law, it's easy to do because the Hardy-Weinberg law states that they are predictable. And you've got your formulas there. I'm not going to run through them. It's pretty self-explanatory. But the idea is the genotype frequency of the dominant homozygote and the recessive homozygote and the heterozygote should all add up to 1 if you've done it correctly. So these, the Hardy-Weinberg law also states that when you get a small group of a population, they migrate out of an area, eventually the genotype frequency is actually going to return to the genotype frequency of the original population. So, and what we're looking at here is we're just looking at these peppered moths, which are traditionally white and black, not orange. But anyway, that's just artistic interpretation. So what we've got here is we've got two population. Well, we've got three groups really. We've got a positive a dominant homozygote, a recessive homozygote, and then a heterozygote. And they've moved into a new area. And what we see here on this right-hand side is these equations being applied. You can review this more in your own time, but you can see that everything will eventually add up to 1, and we're actually able to figure out what's going to happen after a single generation. Things will return to Hardy-Weinberg equilibrium as a result of the equations covered here. Alrighty then, so mutations and genetic disorders. There is 3 main things we need to cover here. The first of which is those sort of point mutations where you get a single nucleotide changing. The other one is where you get an intra-chromosomal mutation, which just means part of chromosomes are just sort of getting mixed around or deleted or messed up. And the other thing to consider is when we get like chromosome level mutations, which means entire chromosomes are getting deleted, they're getting added, or entire haploid numbers are getting multiplied. Okay, so the first thing to consider is the inheritability of these mutations. If you have a mutation happening during mitosis, that is a somatic mutation, which means it's only going to appear in the non-gamete cells that arise from this original mutated cell, and it's not going to be passed on to the next generation. And then they've got germline mutations, which do occur in gamete cells and will be passed on to the next generation. Okay, so phenotypic effects. The first of these is silent mutations, which just means the genetic code did change somehow, but it didn't change enough to change the actual protein that results from that gene. So it's generally pretty okay as a result of the redundancy that we discussed earlier. The other thing we can have is a loss of function mutation, which actually does tend to affect the protein function, or you might have gain of function, which leads to a protein with a different function, but the protein is still ultimately functional. Or you could have a conditional mutation, which just means that there has been some sort of mutation and there is some potential for that mutation to be expressed in the phenotype, but that expression is only going to happen under some very specific circumstances, which are generally rare. Okay, so let's talk about that first level of mutation, just nucleotide level mutations. This just means that you've got a single nucleotide that either gets deleted or it gets replaced or it gets added, and that's going to cause some sort of problem when it comes time to create a protein. The first of these is a silent mutation, which just involves the change of a single nucleotide. Now, this is okay, and you're going to still end up with a perfectly normal a protein, because it's either going to be in the intron or because of the redundancy of the code, you're going to end up with a normal phenotype. You've got missense mutations, which change one nucleotide, and they change one amino acid as a result, and this may affect protein function. You've got nonsense mutations, which change one nucleotide, and they cause a premature stop codon. Do you remember how we talked about how there were 2 or 3 stop codons, and if you get a stop codon occurring halfway through the gene that you want to create a protein from, you're not going to end up with a protein at all. Okay, and then you've got frameshift mutations. Now, this is a problem because if you throw in a new nucleotide somewhere along the gene, it's going to change actually where we recognize the codons to be, and that's going to modify or destroy the entire translation process. An example of one of these point mutations is sickle cell anemia. So sickle cell anemia is a... Yep, let's hear it. Yep. Let me take it. Where are we looking? Appear in non-coding DNA. You mean intro that part? Okay, well, I mean, mutations are occurring randomly, right? So you've got that entire process of mitosis happening, and it's happening with you've got like lots of DNA polymerase showing up to the party, and there's lots of DNA replication occurring. And there are a couple of checks and balances in place to ensure that the DNA that's created is actually representative of the original DNA, but some mutations are going to occur. Now, where those mutations occur is completely random. There's no saying it's not completely random. Like we're studying like colon cancer, where there's like a part where a mutation is more likely to occur, but you might have mutations occurring in exons, which might be expressed, or you might have mutations occurring in introns, which ultimately aren't expressed. It's quite random. What we're hoping for is that the mutations actually do happen in the introns, because these guys aren't expressed. It won't affect the phenotype, and it won't affect protein function. Does that help? Yeah, I don't either. But listen, I'll try and get back to you. Yep.
reason is because the introns, right? So they're being cleaved from the pre-mRNA. So then, of course, if you get mutations there, they're being cleaved, so you get rid of it. So then it won't be expressed. It won't be translated.
Yeah, let's get some. Mm-hmm. Okay. All right. Nice. All right. Is there anything else that anybody wants to say before we can continue? Okay. Look, I know the question you're asking still hasn't been answered yet. You're asking why it occurs in introns. Is that the question? I've claimed that it does, but I haven't explained why. Is that what you're saying? Well, man, it's just the way it is. You've just got to accept it. I'm so sorry. All right. I'm so sorry. All right. But look, I'll try and get back to you. All right. All right. So what we're looking at here is sickle cell anemia, which is okay. Can we have your attention back, please? We've got like less than half an hour left. Okay, so we're going to need to pick up the pace a little bit. Look, sickle cell anemia is a point mutation. All right, so intrachromosome level mutations. Now, this is a situation where you get part of a chromosome being affected by a mutation. You're either getting part of it deleted, you're getting part of it flipped around during the DNA replication process, you're getting homologous chromosome fragments recombining to create incorrect partners, which is called duplication, or you're getting chunks of different chromosomes just inserting into each other wherever they like, not following the actual parameters of a healthy DNA replication, which is called translocation. Now, why do we care? You're going to end up with some sort of condition if you have an intrachromosome level mutation, like thalassemia. So inside that gene that's coding for the hemoglobin protein, you might end up with some sort of dysfunctional hemoglobin if you have a intrachromosome level mutation occurring. Now, depending where the mutation occurs, defines whether it is the alpha chain or the beta chain which is actually affected. Now, mutations, they can be a bad thing or they can be a good thing. So genetic diversity is a good thing because that means the population can withstand or parts of the population rather will be able to withstand new selection pressures, or parts of the, like a disease or something else. And it also might be helpful for evolution. It might offer a selective advantage if you get some sort of new functional proteins that show up. Or it might be harmful because you might end up with some sort of condition like Down syndrome, which we'll discuss in a moment, or they might cause no problem at all, like if it's a silent mutation. All right, so chromosome level abnormalities. Now, these are the big dogs. These are the ones that cause the most problems. So inside the karyotype, which is just your 46 chromosomes, you might end up with an entire chromosome or a couple of chromosomes being abnormal, or you might even end up with a multiplication of the haploid number, which is greater than 2. Now, let's talk about the first type of chromosome level abnormalities, aneuploidy, which just means that you've got a couple of different types of, you've got more or less of a type of a particular number of chromosomes showing up in the resultant cell after cell reproduction. So you can have meiosis non-disjunction or mitosis non-disjunction, which just means that you've got more than the required number of chromosomes showing up in the created cell, or you can end up with monosomy, which just means that there is a loss of a single chromosome of a particular number. You can have trisomy, which is 3, or tetrasomy, which is the gain of 2 chromosomes. Now, the famous examples are listed here. The most common, the one that you're most likely to come across on placement or in a clinic or whatnot, is actually Down syndrome, which is trisomy of the 21st chromosome. So now polyploidy just means that you get an entire, all of 23. So you've got a haploid number of 23, and in your diploid cells, you're meant to have double that, 46. But if you start to have more than that, like 3 times 23, which I can't figure out right now, or 4 times 23, that's going to cause all sorts of problems, right? Generally not compatible with life. Okay, so now when we get these chromosome or intrachromosomal level abnormalities occurring, they can either be balanced, which means there's actually no net loss or gain of genetic material, which means it's not going to be expressed and you'll be fine, or you can have an unbalanced rearrangement, which is going to be problematic due to loss or gain of genetic material. Okay, so structural chromosome level abnormalities. Now, this is a situation where entire chromosomes actually begin to modify their structure in an abnormal way by parts of chromosomes being translocated to areas where they don't actually belong. So the first of these is a Robertsonian translocation, where you get two chromosomes. Now, both of these chromosomes have a long arm at the bottom, or a long arm somewhere, and a short arm somewhere else. Okay, and you get those short arms just running off to do something else. They go AWOL, and these two long arms actually join onto each other, as shown in this diagram here. And this is generally why you get Down syndrome. This generally happens at the 21st chromosome. Now, you could also have reciprocal translocation, which just involves just part of chromosome swapping over, or an insertional translocation, which is very similar to a reciprocal translocation, except you actually get insertion of it into a specific segment within the chromosome, not just tacked onto the end there. All right, so we've made good progress here. We can talk about our final section now, which is DNA. I've sort of rushed because I thought we'd be running out of time, but now we made it. This is good. All right. We'll have time for a couple of questions, which hopefully I'll be able to answer. All right, DNA technique. So the idea here is we now have a very good understanding of how DNA replication and translation and transcription, how it all works, which means we can begin to modify the genetic material in a way that we find helpful. Okay, so the first of these, the first thing you need to understand is that DNA can be cut up, it can be cleaved. How is that done? Well, you get a restriction enzyme, which is actually a microbial defense mechanism. So if there's a virus that shows up and tries to infect a microbe actually has these restriction enzymes lying around where it will actually begin to recognize where there is viral DNA as opposed to its own DNA, or viral genetic material rather, which could be RNA, rather than its own genetic material. It will recognize that and will actually cut the genetic material of the virus so the virus cannot replicate. Now, this can result in a couple of things. It can result in a blunt end, which just means that the restriction enzyme cuts the genetic material straight up and down like that, so that you can't actually get two blunt ends and actually stick them together to create a new, like, cohesive piece of genetic material. But if you get sticky ends, which is just when the cutting is staggered, so it cuts through a phosphodiester bond here, and then it cuts through a few hydrogen bonds, and then it cuts through the other phosphodiester bond on the other side, then you're going to end up with a sticky end, and these ends can actually stick to each other as seen in this diagram here. And why is that important? That's important because if you find a restriction enzyme which is capable of creating sticky ends, you can start cutting up DNA and inserting DNA that you want into the genome of a creature. All right, so analysis of DNA fragments. Now, this was a classic amongst those of us that did biology 3-4. What happens here is we throw a restriction enzyme at some DNA, and it cleaves it wherever it recognizes it's the specific site that it is designed to cleave at. Now, depending on whether the DNA is mutated or not mutated, or is of a particular genotype or another genotype, it will be cleaved at different spots because they'll have different sequences, which means that the restriction enzyme will be working in different areas, and that will result in different sized segments of DNA. So when you throw it inside this gel and you apply some sort of charge, because DNA is negatively charged and it tries to move towards the positive electrode, what that's going to result in is the very shortest pieces of DNA, which are smaller and which are lighter and which can travel through the gel easier, are going to run all the way towards the end here, and the larger pieces are going to be stuck back here. Now, there's a few different purposes of this. It can be used to identify whether a disease. It can be used to identify whether a particular type of DNA manipulation was successful, or it can be used to realize, like, who's your father or whatever. Okay. Now, introducing foreign DNA. Now, okay, so introducing foreign DNA. So now, if we're going to introduce foreign DNA, we're going to need to introduce it into a way that the cell can actually, the nucleus can actually accept that and get it stuck into its own genetic material, right? We can't just throw it and expect it to work. So what we need to do is we need to introduce some sort of vector. Now, this is generally some sort of virus or a liposome, because genetic material doesn't actually just flow in and out of cells. It needs to be traveling in some sort of virus or liposome. Now, inside that virus or liposome, we're going to have a plasmid, which is this circle here. Now, a plasmid needs to contain a couple of things if it's actually going to show up inside our actual genetic material and be expressed. The first of which is a replication unit, so it can actually be propagated and in host cells. The second thing it needs is a site for the restriction enzyme to actually recognize and cleave, so that new DNA that we want to be inserted can actually be inserted into the plasmid. And the other thing it needs is markers. It needs to have some sort of way of recognizing whether the introduction of the foreign DNA was successful. So it needs to be fluorescent, or it can have, like fluorescent, you can literally just see that under a light, a specific type of light, or you can have antibiotic resistance. If the DNA that you introduced that has, if the DNA that you introduced to a microbe codes for some sort of antibiotic resistant protein, what that means is when you throw an antibiotic at a culture of proteins, only the proteins that have successfully introduced the foreign DNA to their genotype are going to survive. Or you can have a color-producing protein inside your plasmid, which is quite obvious. You can just eyeball it to see if it works. Now, transgenic mice. Now, I really struggled with this last year. I distinctly remember that. So let's just break it down. Let's make it as simple as possible. So mice, or particular types of mice, have a gene that codes for a protein. Now, that protein provides the gray pigment that turns their fur gray. Now, if we wanted a mouse that wasn't gray, if we preferred a white mouse for some reason, then what we would need to do is knock out that gene. So there have been scientists that have actually done this, have successfully knocked out a gene and created white mice as opposed to not white mice. That sounds pretty sick, like some sort of experiment at like some Auschwitz or something. But look, don't like get caught up in that. Like, don't think about it too much. Just think about the science, okay? Okay, so you've got a knockout mouse. I've got a gene that produces gray pigment. What do I want to do? I want to stick some sort of random DNA inside that gene and knock out its functionality, so I don't have gray pigment. I'm just left with a white mouse. Now, why would this actually be useful in the real world? We don't want to create white people. What we want to do is we want to realize whether the function, we want to realize exactly what the function of a gene is. We want to realize if our drugs are working given specific genotypes, and we also want to realize what the gene-to-gene and gene-to-protein interactions are, and also protein-to-protein interactions. That isn't written down there, but it's also something worth considering. So how do we get this done? Well, we introduced one of the plasmids that we were talking about earlier, this vector. So it's created by the recombinant technology, which we discussed earlier, and it codes for this new color. Now, it doesn't actually code for a new color. All it does is just some like shambles of DNA that are useless that make the existing color gene dysfunctional. So once that is thrown into the mouse at some point, hopefully it's going to result in that protein no longer being produced. So if we introduce this at a later stage, you're going to end up with a mouse with white patches. So you need to do it earlier than that. You need to do it at the embryonic stage to ensure that this sort of useless gene is actually in every cell of the mouse's body, so that it is entirely white. Okay. All right, and then yeah, so if it's done effectively, you're going to develop an embryo, and birth's going to happen in a healthy way because this is the only gene that it affected if it works properly, and you're going to end up with a transgenic mouse or a knockout mouse. Now, another couple of DNA techniques to consider is the polymerase chain reaction. This is how we replicate DNA in a lab because we can't do it cellularly. So what we do here is there's just a couple of very easy steps. We just denature the DNA with heat. Now remember that this will only break the hydrogen bonds. So it'll just create two strands. It won't break phosphodiester bonds and give us a mess of nucleotides. It will just create those two strands that we want. Now what do? We throw in our prime strand so that the DNA polymerase knows where to work. And then we throw in the DNA polymerase. Well, look, we're not throwing anything in. This is already all in the container, by the way. But what we are doing is changing the heat. So initially, during that first stage, we have high heat so we can denature. The second thing we do is we decrease the heat so that the primer strands can actually get bonded onto the DNA molecule. And the third thing we do is we reapply heat so the DNA polymerase can get to work, because it has a very specific temperature that it likes to work at. Now, after you've done those 3 steps of just modifying the temperature, because everything is already inside the container, you just keep going through that cycle of changing the temperature to get more and more DNA. Each cycle that you do of the polymerase chain reaction will result in double the amount of DNA. So you can just have 2 to the power of however many times you've done this cycle to figure out how many molecules of DNA you're left with at the end. Now, detecting genetic variation. We're going to talk about a couple of techniques about how this is done. But this is basically done when we realize there's a condition that actually has no signature in the bloodstream, so we can't just do a blood test to see if somebody has a condition, or maybe the condition is something that is recessive. Maybe there could be carriers of the condition out there. They don't know about it, and there's no other way of checking whether they're carriers other than doing genetic testing. Or we could be trying to do a prenatal diagnosis, so there's no way of actually testing what's going on inside the fetus other than retrieving some genetic material and running some tests on it. Now, how are these tests done? Well, the first of which is this is gel electrophoresis, but it's being used in a very specific way. So what we've got is we've got a restriction enzyme that is cutting at a specific site that is only present inside the normal genotype, not present inside the sickle cell gene, right? It's only present inside the healthy the normal hemoglobin gene. So if you introduce the restriction enzyme, it's going to cleave the normal gene, but it's not going to cleave a sickle cell gene. So when you put these in gel electrophoresis, you're going to realize you've got 2 strands of the normal gene, but you've only got 1 larger strand of the sickle cell gene, which is going to be stuck further back because it's larger, it's more difficult for it to move. Okay, so other ways of doing DNA testing is just denaturing the DNA into the 2 separate strands and then throwing a probe onto it. Now, the probe can be fluorescently labeled or it can be radioactively labeled. That's not really important. What is important is that the probe is complementary to the specific sequence of DNA that you're looking for. Okay, and then once you're able to eyeball it, or maybe you need to shine a specific light on it or check for its radioactivity, you're able to find out whether specific segments of DNA occur inside somebody's genotype. Now, I believe that this is the final DNA testing method we're going to look at. This is allele-specific PCR, which all this is doing is combining the previous method we talked about, which was the whole method of throwing on a primer, and it's also combining that with the PCR, with the polymerase chain reaction we talked about earlier. So what we do is we denature the DNA, but we throw in 2 primers this time. 1 primer that is only going to bond to the normal gene sequence, and another primer that is only going to bond to the mutated gene sequence. Now, and then we do our polymerase chain reaction. We've got the 3 steps, and we repeat that over and over and over until we've got enough DNA to actually analyze. Now, the results of this will be one of 3 things. You're going to have replication of only the normal DNA. You're only going to have replication with the normal DNA primer, which means the DNA was, of course, all normal, or you're only going to have the mutant primer successfully replicate the DNA, which means that all of the DNA was mutant, or you're going to have both primers work, which means that it was a heterozygote. There was a bit of mutant DNA present, but there was also a bit of normal DNA present, unmuted DNA present. Okay, all right, so diagnosing infectious diseases. Now, of course, this has clinical relevance because you're trying to do HIV tests, you're trying to test for carriers and whatnot. But yeah, I feel like we've covered most of this stuff. Oh yeah, something to consider. With HIV testing, the traditional antibody tests aren't always as accurate as we would prefer. You're going to get false negatives, you're going to get false positives. If you actually take a look at the genetic makeup of the genetic material that just happens to be floating around inside the blood, because the virus will leave some sort of genetic breadcrumbs along its path, you're actually going to be able to figure out whether the virus is present a lot more effectively than just looking for antibodies that the body itself has produced. And now DNA fingerprints, this is what I was talking about before, like you need to find out who your dad is. This is the only way to do it. So what you do is you've got these little short, really variable polymorphic sequences in your DNA. They're hopefully specific to the individual, and hopefully the ones that somebody got from their mother will be different to the ones they got from their father. And then what that's going to allow us to do is, once we use our, restriction enzyme to cut the DNA up in the places that are specific to the short tandem repeat section, or to do the short tandem repeat section, and we throw it into our gel electrophoresis, we're going to end up with these different lines inside the gel, and we're going to be able to realize, okay, so we've got one here that matches mum, one here that matches dad, and they are definitely the parents of this particular offspring. Okay, so practice questions. Now, this whole situation with exams and whatnot is this, in my experience, we love to, in lectures and tutorials, we love to talk about these broad, fantastic, fascinating concepts, and then you show up at the exam and they're testing something very specific, like a really small little fact. Now, every time that happens, it drives me a little closer to the edge. So what I want to do here is I want to cover a couple of the small details that we talked about today, but this does not negate the need for you guys to go home and actually review the entirety of the content. Okay. So, how are we going to do this? Am I going to pick on somebody, or is somebody going to volunteer? Hey, all right, yeah, no, it was easy. Okay, now this is simple stuff. This is still pretty simple stuff, just a bit of maths. Make sure you realize, nicely done. Okay, this might take a little bit more time, hopefully. All right, figure it out. Give me a shout when you know. I can't even remember the answer. What do we still working on? B? B? Okay, well, this is going to be a surprise to me as well. It's C. So sorry. Okay. Well, look, when you go home and review this stuff, actually take a look at how this is done and try and figure out some sort of algorithm that you can use to make sure you don't get this wrong in the future. And now remember also that you need to consider that there are stop and start codons as well. Like it doesn't follow logically that there's going to be you're immediately going to start with or you're going to get a tyrosine rather than a stop codon. You need to actually figure it out and see which sequence specifically matches the amino acid sequence the question is asking about. Okay, now this is just, there's more than one correct answer for this, so we're looking for two correct options here. Sorry? Where am I looking? Oh yeah, so sorry. So what do we feel? What are our two correct answers? Can anybody specifically answer the question without like a whole lot of murmuring that I can't understand down here? Say anything again? A and D? Nicely done. A and D, correct. Okay, so C, yeah, I'm getting a general vibe of C, nicely done. Now, I sort of rushed over a couple of these. We were pretty clear about Down syndrome. What do we feel the answer is? F, nicely done. All right, sickle cell anemia. This is the one that I didn't cover this. Sorry? B, all right, nicely done. So that's the entirety of genetics. You've covered it, nicely done. All right. Now, back to slide 40? Yeah. Right. Is there a mistake?
No, you're correct. They made it.
40?
Oh,
that's
that's it. Yeah, one more. Just really quickly, to clarify this for anyone who's watching the recording, Abe actually wasn't incorrect. What he was listing here is the two different types of ways that you can have a silent mutation. Number one is redundancy of a genetic code. As we know, we have multiple codons coding for the same type of amino acid. So redundancy is one. Secondly, the second instance in which you can have silent mutations is when you have a mutation occurring in a non-coding section of DNA. Not specifically introns, but introns is one of the examples. That's all he was talking about. He wasn't mentioning that silent mutations can only occur in introns. Obviously, they can occur in exons as well. But other than that, I think that brings us to the conclusion. Thank you very much, Abe.
No worries. What the hell? What are you doing?
I rewatched the recording just to make sure. Whenever you're ready to start. You have the thing already up there. Yeah, so you might just need to log into. Just go to your Google. My Google. Or else you have a USB. I have it on my laptop. I'll log in. How's it been today? It's been alright. Yeah. That's all the presenters were. Yeah, they were pretty good. Yeah. What's that? Oh my god. So this will amplify, this one records. Okay. I'll get it up to you and then I'll show you. Is it in the post lights? Yeah, it's in the post. Which one? I do remember the holidays, so it's fine. I like it. This stuff would be like, Hey guys, how you going? Hi. So just before we start, I'm not sure if this will be possible for all of you, but does anyone want to stand up for me? Yes. Yeah. Stand up. Stand up. Thank you. And I'll get all of you to just raise your hands above your head as high as possible. And I'll get you to high five the person next to you. And just yell your name really loud for me. Yeah. All right, great. Thanks, guys. Just take a seat. All right, thanks, guys. So today I'll be presenting neurophysiology. I think it's a fantastic subject, which is unfortunately taught very poorly. So hopefully, if you guys already get it, that's great. This will just be some revision. If you guys, like me, this time last year, have no idea what's going on, that's okay. A lot of the stuff the faculty teaches you is quite dumb. We'll skip over that. We'll try and get you the stuff that's important, the stuff that's really going to come up next year and in next semester. So there's my name down there. So if you have any questions and you don't want to put your hand up and ask because you think that's weird, that's okay. Just send me a Facebook message and I'll check them at the end and I'll just go through them. So what am I going through? I'm going through neuroembryology and neuroanatomy very quickly because this sort of shit will not probably come up on your will not probably come up in the future, but it might come up on your exam. And then I'll go through neurophysiology in a bit more depth. Is that cool with everyone? Yes. And I'll just get Quran, just because you're right in front of me. If I say something that hasn't come up in any of your lectures, just give me one of these, just so I know I can skip over that, or I can do that in more detail. And if something you think needs more detail, just give me one of these or something. Yeah, thanks, Quran. All good. Any questions before I start?
Hopefully not. Great. So neuroembryology. You guys did get a lecture on this? Yes? No? You didn't get a lecture on it? Okay, cool. All right. So neuroembryology, I don't know why you get a lecture on it. It's kind of really dumb, but it leads to a few important clinical stuff. So, yeah. The lecturer, who did you have? Did you guys have Paul McMahon? Cool. Great. So what we should already know from embryology, we have gastrulation, which has already occurred, and we have a trilaminar disc. Hopefully this sounds familiar. If not, you can just revise. It's quite simple. You've got the ectoderm, mesoderm, endoderm, yeah? Hopefully you know what that is. You've got a picture in your mind what that looks like. Yeah, it looks like a sandwich with the mesoderm in the middle. So if you know that, you then know what's the basis for neuroembryology. This is a fantastic table to memorize. Unfortunately, that's the only thing you can do with it is memorize it. But if you know, especially the ectoderm, just kind of memorize these for neuroembryology specifically. You'll be fine. So I'm going to go through this really quickly. Please put your hand up if you want me to go through it in a bit more detail. Also, sorry, I talk really fast. That's just how I am. So if I am too fast, just put your hand up and ask me to slow down. That's fine. Cool. So neural induction. So I'm going to go through it in 3 steps, 3 slides. Very simple. Yeah. So you have this thing called the notochord. Yeah. The notochord is a structure that exists in the mesoderm. It does a lot of things in embryology, but for now, and even now, like up to us, all you need to know about the notochord is that it's sort of signaling for the starting of neuroembryology. Yeah, so the notochord signals the ectoderm above it, because the notochord's in the mesoderm. It signals the ectoderm above it to form a neural plate. So what's the neural plate? It's just a thickening of the ectoderm. Yeah, so this sort of tissue differentiates from the rest of the ectoderm, and we call it the neuroectoderm. Yeah, very simple. Not going to go into too much detail. So then the neural plate, yeah, because it's a plate here, it forms a groove, and now it's called the neural groove. Very simple. So the neural groove keeps on pinching, keeps on folding until it sort of pinches off to make a tube, and at this point it's called the neural tube. Very simple. So now you have the ectoderm as a layer above, and you've got this tube of ectoderm or neuroectoderm in the mesodermal area, but it's still ectodermal cells. Does that make sense? Is that cool with everyone? Cool. So yeah, now you have the neural tube. Also, you have neural crest cells. So not all of the tube kind of pinches off. There's a bit on the side which kind of detach and come on either side of the neural tube. Now in the face, because remember you've got this embryo that's looking like a sausage, and this sort of entire process is happening all along the sausage. So the sausage means the head on one end and the tail on the other end. So towards the head end of the sausage, these side bits are going to make the connective tissue, melanocytes, which give you pigmentation, that sort of shit. And towards the torso area of the sausage baby thingy, they're going to become the ganglions and the peripheral neural tissue. Because if you remember, only ectoderm can form nervous cells. So the nerves in your fingers, because you have nerves in your fingers to move your fingers, they also come from these side bits called the neural crest cells. So that's basically it. That's all you really need to know for neuroembryology. Any questions before I move on? Yes. Sure. So if you have a look here, the purple bit, so the purple and green bit is all neuroectoderm, but only the purple bit will form the neural tube. Now the green bits will come off here, and then they'll slide off to the sides, and that's the neural crest cell. Does that make sense? Cool, great. Just a quick clinical note. This is really the only reason why this is important to you guys. I'm going to keep bringing it back to clinical stuff, and maybe at this point you won't really know many clinical things, but that's fine. Hopefully you can build upon this for next sem. But the clinical note is that if the neural tube doesn't close, because as you remember, it pinches off and closes. If it doesn't close rostrally, which is towards the head, you can get anencephaly, which is this sort of thing. And if it doesn't close caudally, you can get spina bifida. Did you guys have a PBL on spina bifida? No. Wow. All right, we had a PBL. Oh wait, sorry, that's the next sem. You'll get that next sem. Sorry, yeah. So that's basically what spina bifida is. So how do you remember it? Rostrally, round, your head is round. Great, fantastic. moving on. If you have any questions, feel free to message me on Facebook. I'll check them at the end. Neuroanatomy, really quick once again, because I kind of really want to focus on physiology. Physiology is really fun. So I'm going to spend a bit of extra time on this slide just because it'll come up again, and it's something people don't often get right first time, and it's not your fault, it's the way it's taught to you. That's whose fault it is, yeah? So you've got the nervous system, and it's important to remember that there's two parts of the nervous system. The first part is the brain and spinal cord, the central nervous system. Now, I can't really tell you about this because I don't know a thing about it yet. I'm going to learn about it next time as well. But the peripheral nervous system is something you're going to learn about a lot next semester. When you do upper limb and lower limb, that's all peripheral nervous system, yeah? That's some of the really heavy stuff. And then there's also peripheral nervous system in the abdomen and everywhere else that you also do next year. So the peripheral nervous system is made up of motor neurons and sensory neurons. This should make sense, yeah? This should be quite, like, you know, when you're a kid type biology, yeah? So motor neurons, I move my fingers. Sensory neurons, I touch things, yeah? So they're very different and they require different neurons. Just a thing about sensory neurons, with the table, I touch the table and I can feel that I'm touching the table. But you also have sensory neurons like in your stomach, yeah? Because when you get full, your stomach stretches. Even though you don't feel like the stretching itself, but you do feel the feeling of fullness. Yeah, these are still sensory neurons. Yeah, that's just something that'll come up later. Don't stress too much about this slide. If you're one of those people that are just cramming to pass the exam, that's fine, that's respectable. Don't worry too much about slides like this. I'll tell you which slides not to worry about too much. Then you have motor neurons. Now, I'll talk about somatic motor neurons first. This is very easy. You have stuff like moving your fingers, moving your hands, yeah, that sort of stuff. All of that you can feel that you're doing. Very simple. Somatic, that's just what somatic means. Then you have the autonomic nervous system. You guys have heard of the autonomic nervous system before? Yes, fantastic. So this is stuff that is involuntary. But as you can see, the autonomic is under the branch of motor. So if someone tells you, and people will tell you this, if someone tells you, oh, the stomach is like, you know, parasympathetically innervated and it tells you when you're full, that's wrong. Because parasympathetic and sympathetic are purely motor, yeah? So constriction of arterioles, constriction of bronchi, the lungs, you know, that sort of stuff is all, like, movement of things is all autonomic, but sensation of things is not autonomic at all. And you'll see websites, you'll see like actual like places that look legitimate sources that will say this. Just be careful. So sympathetic and parasympathetic divisions. So these are the two divisions of the autonomic nervous system, and the division is purely anatomical, yeah? So you've got the sympathetic nervous system in the middle of your body. So your thorax, it's sometimes called the paravertebral system because it's on either side of the vertebral column. Then you've got the parasympathetic, which is like your head and your neck. It's sometimes called the craniosacral, because cranium and sacrum is the bone that makes the booty. So the sympathetic nervous system is the fight or flight. Very easy way to remember it. So if you are about to be attacked, what things need to happen in your body for you to survive? Yeah, your heart rate is going to increase. That's sympathetic. The blood flow to your organs, like your stomach and your digestive system, are going to constrict, and the blood flow to your arms and your legs, so you can run away or beat up the thing, are going to dilate, so your muscles get more blood. Yeah. So sympathetic nervous system controls all of blood everywhere. Yeah. Then your pupils dilate. That's also sympathetic. Your bronchioles, which are the passageway for your lungs, they dilate as well, so that more air can get into your lungs. That's all sympathetic. It's very easy if you remember parasympathetic and you remember fight or flight. You can kind of derive all the functions. Then you have the, sorry, I said sympathetic, yeah, cool. Then you have parasympathetic, and this function is mainly rest and digest. So the parasympathetic is very, you know, digestion of your food, that sort of stuff. So yeah, they have the opposite functions generally, and you can remember that sometimes they work with the sympathetic nervous system. So for example, that's wrong, sorry, I'll fix that. Sometimes they can work with it, so erection and ejaculation, and then sometimes they can work against it, such as dilation or constriction of the pupils or the bronchi. It wasn't wrong. So yeah, thank your dad's sympathetic nervous system and parasympathetic nervous system. Fantastic. So just general definitions that you can know. They're not that important, but next sem they will be kind of, you know, spat out at you randomly. Someone will just say plexus of nerves, yeah? And it's kind of good that you know what that means. And if you don't know what that means, it's fine. You can Google it. Don't stress. But a plexus is a mixing of nerves. It can also be a mixing of anything, but generally a mixing of nerves. A synapse is a gap between neurons, between nerves. Soma is the body of a neuron. A ganglion is a collection of neuron cell bodies. I'll come back to a ganglion and give you an example later. A ramus is a branch connecting mixed sensory and motor fibers. A ramus is also just a branch sort of thing, and that'll come back in anatomy. A root, I'll go through this later. Don't stress about ganglion, ramus, root too much, but they're there. If you ever want to learn them, you can look back at these slides. Other parts of a neuron: axon, dendrite, axon terminal. Hopefully you should know. Axon hillock, that's where the body meets the axon. That's the axon hillock. Wow. So that's also important in neurophysiology. Myelin sheath is like an insulating cover. Node of Ranvier, that's like these little gaps in the myelin sheath. I'm going to come back to their function later, but these slides are just there if you want to go over them later. Great. Layers of a nerve. This wasn't taught to us, but some crazy second year told us to learn them. So what I'm going to do, if this wasn't taught to you, is I'm going to say don't learn them, but they're there, yeah, if you want to learn them. Cool. Glial cells, things that these actually are pretty good. So things you probably should know. These are just other nervous cells, yeah. So they do stuff. They help the nervous system, but they don't actually do the neurophysiology. They don't conduct a current or send messages or anything. So you have oligodendrocytes and Schwann cells, and these are the things that provide the myelin sheath. So the oligodendrocytes are in the central nervous system, and the Schwann cells are in the peripheral nervous system. So how I remember it, Schwann sounds like a nice girl, so you would want her on your PNS. So that's how I remember it. Astrocytes, these are found only in the central nervous system. I'll give you a second. Fantastic. So astrocytes are found only in the central nervous system, and they do a lot of things. Yeah, don't necessarily learn what they do, but like they regulate the ion concentration, they provide energy, they maintain the space. They're just very, very supportive cells. So the way I remember it is astro, think space, so they hold the space between them. Think stars making energy, so they have glycogen, and think being central, because they're in the center of this solar system, and they're also in the central nervous system. That's how I remember them. I learn a lot through mnemonics and dumb stuff like that, so I'll try to give you as many as I can as I go. Great, neurophysiology. Before I continue on to the main part of this lecture, any questions? Great. So the resting membrane potential and the actin potential. So let's just take a second here, because if you don't understand this slide, a lot of neurophysiology won't make sense. So the really cool thing about neurophysiology is that it all just kind of happens naturally. So if someone tells you this channel opens or that happens, by knowing the resting membrane potential, you should be able to figure out what's sort of happening, yeah? So it's kind of a throwback to osmolarity lecture. The osmolarity lecture is very relevant for neurophysiology and renal physiology. So basically, this is what you kind of need to know, yeah? Outside the cell, you have Na and Cl, and you have calcium. Inside the cell, you have potassium, you have phosphate ions, but don't worry about them. They're not relevant to you at this point, at least, or me. And then you have a lot of negatively charged proteins, yeah? So the negatively charged proteins is what holds a lot of the potassium inside. So the way I remember it is that when potassium and sodium come to the cell's like, it's okay, you can come in potassium, okay? And then you say, nah, you're not allowed in sodium, and sodium stays out. And then obviously chloride will always follow sodium because salt, so sodium and chloride are outside. Something that you'll just have to memorize is calcium is outside, and that's because calcium is very, very, very useful in all the cells, and it does things, yeah? But unfortunately, like, it does very special things. Unfortunately, you don't want all these special things happening all the time, and therefore you keep all your calcium outside until you need it, and we'll talk about when you need it later. So a lot of negative proteins inside the cell makes it negatively charged. It's about negative 70 millivolts, yeah, at rest. Now, it's important to remember that there's a charge difference, but not an osmolarity difference, yeah? So the inside of the cell is a lot more negative than the extracellular fluid, but the inside and outside of the cell has the same amount of osmoles, yes? Okay. Point clarity, are you talking about nerve cells or all cells? All cells, but especially nerve cells. Yeah. So all cells are, especially with the sodium and potassium and calcium and negatively charged proteins, that's for all cells, but the negative 70 millivolts, I wouldn't know about the other cells. But yeah, generally most cells follow are very similar because they're all just kind of got very similar structures. Obviously, some cells are unique to their function and they won't follow this. Good question. I had chocolate and I was going to hand it out for good questions and answering questions. So there you go. Sorry, poor effort by me. Great. So obviously now you can extrapolate from this, yeah? You've got a lot of positive things outside the cell, positive things being sodium, but they're not being let into the cell. So the sodium wants to come in, but it can't, yeah? It can't because there's no channels for the sodium. And if you go back to biology or like one of your first lecturers, you remember that the lipid bilayer is lipid, and therefore it does not like passing charged particles through it. Yeah, so for charged particles, you need channels, and I'll talk about channels now. So there's channels and there's pumps. Now, what's the difference? So channels are for passive transport, and therefore you have no ATP required. This is an example of a channel. Once the channel opens, the ions will move from high concentration to low concentration, because all things in nature, all things in physics want to go back to equilibrium. Yeah, so that makes sense, hopefully. And you have voltage-gated channels, which I'll talk about later as well. Obviously, if you had high ion concentrations here and low ion concentrations here, but you wanted to make it even higher, you could not use a channel, because channels are purely through the laws of nature. It's just opening the door and then it's leaving it, and you let whatever happens happen. Yeah, that's why you have pumps. Now, pumps require ATP, which is what this means, yeah? And pumps can do whatever you want them to, because they use energy, and therefore, like when you put in energy, you can make things go away from equilibrium. So they can go against the concentration gradient, but obviously they're slower, yeah? This is like a concentration gradient is sometimes described as like a hill, and you leave a tennis ball on top of the hill, and you put your hand there, and when you remove your hand, it'll go down the concentration gradient. But to get the tennis ball and move it up the hill, you need to physically move the tennis ball. That's energy that you're putting in. And therefore, ATP is required for pumps to make things go against the concentration gradient. And an example, hopefully, that you're familiar with is the sodium-potassium pump, which is used everywhere in the body. And that's what maintains that sodium outside the cell and potassium inside the cell. Fantastic. Any questions up till now? Great. So action potential. This is the real meat of the stuff. And if you understand this, you should be fine. So an action potential is a rapid changing of the cell membrane potential. And this can be used to send an electrical impulse. So obviously, this is where it is. And then after the action potential, it's very positive. Yeah, so an action potential changes the negative/positiveness of the cell at that point of time. So think of it as a Mexican wave, yeah? So you have a row of people. This is one axon. If one person stands up there, that's the positiveness there. And then that passes on, and the next person stands up, and the next person stands up. And that sort of sends that impulse towards the end. So how do we get from the brain to like the finger? For I want to twitch my finger, I send an impulse at my brain. Obviously, there's other neurons, don't worry about that. I send an impulse at my brain that sort of sends an impulse at the start. That impulse kind of bumps all along the neuron towards the end until my finger can contract. Yeah, so that's an action potential. To learn the details of an action potential, which I highly suggest you do, probably because it's also examinable, we'll go through them now. It's best learned in steps, so I'll go through them in steps. So the Nernst equation is something, hopefully, I looked through your slides, and there was a lot of Nernst equation in your slides. No doctor or medical student will ever know the Nernst equation. If you're really into physics or really like have a lot of spare time, feel free to go through it, but I'm not going to talk about it because it really didn't come up anywhere on our exam. It was in your slides? Okay, is that what you were told? Oh, cool. All right, great. So just before I go on to action potentials, you kind of need to know about voltage-gated sodium channels. So an NAV, an NAV is a sodium V, and the V is voltage-gated. So there's voltage-gated sodiums, potassiums, calciums, whatever. But it's basically a channel that opens when the cell reaches a certain potential, usually during depolarization, or it can be any time, yeah? And the thing about these channels is that they're obviously not thinking, yeah? A bunch of little atoms is not like sentient enough to be like, I'm gonna open now to send this message. So what it does is when the cell reaches that potential, when it reaches negative 55 millivolts, which we'll talk about, it just opens, yeah? It's gated by the voltage. Once the voltage reaches something it likes, it'll open that gate. Very simple. The channels have 3 states: open, closed, very simple. Open means it's open, closed means it's closed, fantastic. And then there's an inactivated state. And this is very important, and I'll talk about why this is important later, but just think about it as in like, for a brief period after it's open, it can't respond to a new signal, and it has to remain closed. And this is extremely important, and I'll talk about why that's important. Great. So before I go into the steps of an action potential, are there any questions? Because if you have any questions about any of that previous stuff, you might not understand this. All right, great. So step one, the neuron will receive a stimulus, and this stimulus will open some of the sodium channels. Yeah. So what can that stimulus be? We'll talk about what that stimulus can be in terms of neurotransmitters later. But for example, in your finger, that you have like pressure little like channels that are gated by pressure. So when you touch them, it sort of lets the sodium in to that neuron right at the tip of your finger, and that causes a cascade all along to your brain, and that's how you feel the touch. Yeah. So a lot of them are pressure gated when it comes to sensory information, or stretch gated, such as in your stomach and your feeling of the stomach. Great. That's the stimulus. Yeah. And now once there is enough stimulus to make the membrane about negative 55 millivolts, that's when the action potential starts. Now the reason of this is because all those sodium gated, voltage gated sodium channels are going to open when the channel hits about negative 55 millivolts. Negative 55 millivolts. And at that point, all of those channels are like, yep, this is the signal I need for all of us to open, and they all open. Now, naturally, knowing osmolarity and concentration gradients, you know that there's a lot of sodium outside the cell and like very little inside the cell. So it kind of wants to get into the cell. So if those channels are opened by this like negative 55 millivolts, obviously all the sodium from outside the cell is going to rush into the cell. Yes, does that make sense? Hopefully that makes sense. Yeah, any issues with what I've said so far? Any mistakes I've made that you've heard? All right, great. So once all those sodium rush into the cell, sodium being positive is going to make the cell what? Also positive. So yeah, the influx is because there's like a reduction in resistance. So there's a lot of physics behind this, because obviously like biology and chemistry are the real subjects that are really relevant to everyone. But like to really understand them, you have to go down to a physics level. The physics level is not required for the course, but it's very interesting. So if you are like into that, go for it. So once the membrane potential has reached its highest point, or at 30 millivolts, this is what signals the potassium channels to open. So they're also voltage gated, and they open. And obviously, potassium is inside the cell in a high concentration, so it wants to flow out. And that's what happens. The potassium flows out of the cell, and suddenly the cell, from being very positive, goes back down to a negative level. Hopefully that makes sense. Now, a thing with the potassium is that more potassium goes out than is really necessary. And this means that the potassium overshoots, so it goes way more negative than necessary. Now, this ensures that another action potential isn't fired back in the direction that it came from, yeah? Because there's that little lag period. One action potential is sent, and then there's a pause, and then there's another action potential sent. Then you obviously have a restoration of the resting membrane potential by the pumps. Now, sorry for this being very text-heavy. Now I have this as well, and I'm just going to quickly go over it again. The reason I did this is because some people like learning like this, some people like learning like this, some people like learning like this, some people like putting it together. So I'm just going to go through it very, very quickly again, and this is there, and you can message me if you have any issues with this. This is simultaneously very complicated, but once you get it, you're like, oh shit, that was so easy. Why did that take me so long? So that's fine. It might take you a while. It might take you a few read-throughs. Those videos, those crash course videos are fantastic for this as well. So basically, your cell's at rest. It gets some impulse. It reaches that gate threshold at negative 55. All the sodium channels open. There's depolarization because all the sodium runs in. Sodium being positive makes the cell positive. At about here, the sodium channels close. The potassium channels open. And this is not because they want to open. It's because it's in their nature. That's how they're designed. At that voltage, they will open. The sodium channels, sorry, the potassium channels will open. All the potassium will run outside of the cell. And suddenly you've got a very negative cell. And then you've got, this is called hyperpolarization. And then you have the sodium-potassium pumps, bringing it back to the resting membrane potential. Any questions? All right, fantastic. This is all there. If you are complicated, go through it. Refractory period. This is very important. So there's two types of refractory periods. So you have a relative refractory period, and this is, as I talked about, the hyperpolarization state. So obviously, let's think about the same example I gave before. When I touch my finger, when something touches my finger, there's a little impulse and a few sodium ions run into the cell, yeah? Now, at rest, at negative 70 millivolts, you only need 15 millivolts of sodium. Oh, sorry, you just need a 15 millivolt increase for the action potential to be triggered, yeah? So a small touch will trigger it. At this point, because it's being, there's like hyperpolarization, you need a much harder impulse, yeah? And what this does is it just stops a lot of impulses kind of going at once. But what it also means is that if you need a lot of impulses going at once, for example, when you're like, lifting a really heavy weight and you need all of your muscles to contract, that's when this can kind of be overturned, where you have a lot of stimulus and it doesn't matter. It's going to make it up and it's going to go all the way back up and have another action potential. That's a relative refractory period. It's relative because it can be overcome. Then you have an absolute refractory period. This is absolute because this is when the nerve will not respond to any more signals. So the absolute refractory period is about here, yeah? It's about that area. And the reason you have this absolute refractory period is because those sodium channels are inactivated. So because the sodium channels want to open at negative 55 millivolts, the issue would be during depolarization, it would hit negative 55 millivolts again, and it would send another action potential if there wasn't for those inactivating states of the sodium. Hopefully that makes sense. It's a bit complicated, but if you think about it and look at some videos, that should make sense. So because it's inactivated, those sodium channels just aren't going to open until you reach about here. And once you reach about here, that's when another action potential can be sent. Otherwise, you just have constant action potentials being sent back and forth down the cell because there'd be nothing to sort of stop that excitation. Hopefully that makes sense. Great. So I thought I would break up the lecture at this point and just sort of take a break, as in I'll do an exercise with you guys just to sort of explain this scenario. So there's this thing called myelin. There's myelin sheets. Myelin is a conductor of the axon. Yep. So there's sort of two scenarios. Yeah. So obviously in evolution, it wasn't like, oh, which one should we pick? It was both sort of happened, and one was really good for survival. So there's two options. One is that you have channels along the entire axon and no myelin. Yeah. So along the whole axon, you have a bunch of sodium like gates, and that action potential can kind of occur. And the other option is that you coat the whole cell in myelin, and me at the start and someone at the end is a channel, and you would just kind of hope that the channel will reach all the way to the end. Yeah. So obviously both of these have very like issues. This is an active process and it's very slow, yeah, because it's got to go from channel to channel to channel to channel. But it is accurate. The signal will always reach the end. That's an advantage of this. Now with this, it'll be really fast because it's just going from one channel to the end. The only issue with this is that the signal might not reach there all the time. So which channel is better? Let's do an experiment. On this side of the class, I'm going to pretend that we're all channels, and we're all going to pass this chocolate down to the end. And on this side of the class, I'm going to pretend that I'm a channel and someone's a channel at the end. And we'll see the positives and negatives of both, yeah? So just turn around and pass it to the person next to you. Pass it into their hand. Yeah, cool. So, as you can see, it's taking a bit of time, but you know someone at the end is definitely guaranteed that chocolate. Yeah, that's the advantage of that. Now with this scenario, can I get a volunteer at the back? Cool. Yeah. So I'm a channel, you're a channel, and I'm gonna try and pass this message all the way to the end. So as you can see, I did that on purpose. As you can see, the channel doesn't really reach the end, and that's an issue. So what do you do? Like, which scenario is better? We use both. And the way we do that is we have this thing called saltatory conduction. So basically, it's you have these like gaps in the myelin sheath, and we call them nodes of Ranvier. Yeah, I don't know why we call them nodes of Ranvier, probably because Ranvier named them, but that's not important. And it allows for the signal to jump from each gap in the myelin sheath. And what it does is it guarantees that speed, but it also minimizes energy expenditure, and you're guaranteed that the signal is going to reach the end. So for example, if I threw this chocolate halfway, and then from halfway they threw it to the end, you could guarantee that it would reach the end and it would be very fast. Hopefully that makes sense. That's just something the faculty really emphasized for us, and it's something I didn't really understand first, so that's why I did the demonstration. Is that cool with everyone? Any questions on that? All right, great. So ion channel locations, something you can read over, something you can memorize if you want. It could come up. Well, the sodium channels are kind of everywhere, especially in the nodes of Ranvier. The potassium channels are for repolarization and the resting membrane, so they're kind of everywhere except the nodes of Ranvier. And then the calcium channels are right at the end, and they're important for neurotransmitter release. So I'll talk about them soon as well. Cool. So neurotransmission. Neurotransmission is fantastic because it leads on to a lot of pharmacology. Because as you can expect, a lot of the things in your body happen via your nervous system controlling it, and we can control what happens in the nervous system by controlling neurotransmission. It's actually very interesting, and it's something that will come up later as well. So basically what you're doing is you're converting an electrical stimulus to a chemical stimulus. So in neurotransmission, it allows the neurons to communicate, yeah? Because as I said, that entire axon is one cell. So that changing in the potential, the increase and decrease of potassium, sodium, whatever, that happens because it's one cell. The issue is, how do we go from one nerve cell to another nerve cell? Obviously, we need something to bridge the gap. So what we do is we have neurotransmission. Now, the neurotransmission can be like kind of wherever. Mainly it's from the axon of one cell to the dendrite of another cell. And once again, I like learning it in steps. That's just how my mind figures it. So step one, the release of the neurotransmitters. So obviously before the action potential arrives, the neurotransmitters are usually stored in a vesicle, yeah? So your cells know their job is to send messages. So they just have a bunch of neurotransmitters ready to go for an action potential to come. So what happens is the action potential arrives at the axon terminal. So if we know that this is the terminal of the axon, this is probably where the cell body is, the action potential comes down, 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 down. And here what happens is right at the end of the cell, you have specialized channels called calcium-gated, voltage-gated calcium channels. Sorry, I keep doing that. So what they do is they trigger the snare protein. Now, do you need to know that they're called snare proteins? Probably not. But what they do is they trigger these proteins, and the job of these proteins is, say you have like a vesicle of neurotransmitter, it kind of like snaps it into place. It snaps it, and the two membranes sort of fuse, and all of that neurotransmitter is released outside the cell. It's released into that synapse area. Yeah. So then you have part two, yeah? The uptake of the neurotransmitters. So those neurotransmitters are just there, and they will naturally want to bind with receptors that are just around. So the neurotransmitters will bind with receptors at the next neuron. So what they do is they cause a conformational change. Now, this is something that's brought up a lot. What is a conformational change? Basically, it happens a lot in cell signaling. When something touches a receptor, that receptor changes shape, and the change in shape is what causes the effect of something else. So the conformational change in this case can start another action potential. So obviously the neurotransmitter can do many different things. For example, on your muscles, they do very different things. They cause contraction compared to another nerve. They cause another action potential. Great. Then you either have an excitatory or inhibitory postsynaptic potential. So the E and the I, PSP. So let's talk about both of them individually. So an excitatory postsynaptic potential, very simple. It's just causing temporary to mild depolarization. So as you hopefully remember, because I just went through it, once you get to negative 55 millivolts, you're going to send another action potential. These might not necessarily cause the cell to go to negative 55, but maybe it'll go from negative 70 to negative 60. Or maybe it will go from negative 70 to negative 55. What they do is they just cause a mild increase, a mild excitation. Yeah, and these can add up. So say 3 neurons are sending a signal to 1 neuron, and they're all sort of exciting it. That neuron should hopefully be excited enough to send a message. Also, you can have an inhibitory postsynaptic potential, and this makes this neuron less likely to fire an action potential. It hyperpolarizes it, which hopefully you remember is this part, hyperpolarizes it. And what that means is it makes it harder for another stimulus to cause an action potential. So these can also have an additive effect. So say 3 neurons are sending an excitatory message to another neuron, another 3 can send an inhibitory message to that neuron, and that will cause you to not have that movement, or more likely, this is sort of the really complex stuff happening in your brain, and it'll cause you to not have that thought. Well, hopefully that makes sense. Obviously, at the end, you need to deactivate the neurotransmitters. So the neurotransmitters, if they're left there, they will just keep doing their job because they're not sentient. They're just molecules that excite other molecules. So you can degrade them by other enzymes. So for example, acetylcholine is broken down by acetylcholinesterase. They can bind to receptors on the presynaptic cell. This is this cell, which will inhibit any more to be released. This happens in a lot of autonomic stuff. They can just be reuptaken by this neuron and then repackaged for the next signal, or they can just be cleared away by astrocytes. And many drugs interfere with this. And the reason why it's important is that you can have drugs that degrade the neurotransmitters, or that stop the degrading of neurotransmitters. And therefore, they'll cause a bunch of different effects. And the synapse is fantastic for pharmacology because you can do so much stuff just in that little synapse, and it can have effects all across your body and things that you couldn't even imagine. So as I said, acetylcholinesterase is something that breaks down acetylcholine in the synapse. And I think you have a drug that you were supposed to learn called pyridostigmine, and that sort of stops the acetylcholinesterase. And by stopping acetylcholinesterase, you have a lot of acetylcholine just hanging around in the synapse, causing excitation. Great. So summation, this is something I already kind of touched on, but it's when multiple excitatory potentials occur, and this allows the cell to reach the potential. Now you can have temporal summation, where they're fired one after the other. So temporal, like time, like right after each other in time, or you can have spatial summation, where like two different neurons will excite it at different spots, but the overall excitation will be enough to cause an action potential. Great. Then you can also have inhibition. So once again, that can be pre or post-synaptic. I'm just going to leave this slide here. I mean, it's a really nitpicky term, which I mean shouldn't really matter, but yeah, it can cause signals to not be sent and another action potential to not occur. So just I got a lot of just free loose slides that don't sort of fit anywhere because that's just kind of how Parko teaches. But the axon hillock is a place where all these graded potentials, so all these inhibitory or excitatory sort of add up, and that's when it will either become an action potential or not become an action potential. And there's a very high concentration of sodium, voltage-gated sodium channels here. Great. So neurotransmitters, big scary list. Don't worry about it too much. I mean, they shouldn't really ask you it, but they can. And by the end of next year, hopefully, or like by the start of next year-ish, you should hopefully know these. The way I remember it, or the way I remembered it, especially for last year, where I didn't really want to learn all of them, is that remember acetylcholine and adrenaline and noradrenaline are autonomic, and they cause different effects depending on the receptor that they target. So don't worry too much about them. They do both excitation and inhibition. But for the rest, all of them that start with G are inhibitory except glutamate, and that's because glutamate is your mate, and he is excitatory. Also, dopamine is inhibitory, but serotonin is inhibitory, but hopefully you know that just from depression and stuff. There's a lot of serotonin stuff. So should you learn this? Probably not if you're just trying to pass, but I mean, it's good to just look over it if you're scared. So types of receptors. You have ionotropic and metabotropic receptors. This is something that's quite important, maybe not in neurophysiology, but just in general, like physiology. You have ionotropic channels which let ions in. So once a neurotransmitter binds to the ionotropic channels, they just kind of open and they let ions flow through. So a lot of the channels which are involved in neurotransmission are ionotropic channels. Then you have metabotropic channels, and this is they're more interesting. They do a lot of really complicated stuff, and that's stuff you're going to learn about next year with the endocrine system. But they take longer to act, and basically the neurotransmitter binds, it kind of changes it kind of changes another thing, it kind of changes another thing, and there's an entire cascade within the cell of things that occur, and ultimately this can do something. Now this something can be very different in all cells. For the purposes of neurotransmission, you can also have metabotropic channels which ultimately lead in channels, metabotropic channels which ultimately lead in another channel opening and allowing ions to flow through. An example of a metabotropic channel that you would have heard of is like insulin and glucagon work on metabotropic channels, and then they cause inside cells like transcription and translation, and basically they work on the protein kinases or whatever. You'll learn about this next year, don't stress, and it'll either cause more glucose, like gluconeogenesis, or glycogenesis or whatever. Cool. Any questions so far? No? Great. So NMDA versus AMPA receptors. Did Parker mention this? No. Okay. Parkinson? Did you guys not have Parkinson? Helena Parkinson. Oh, okay. All right. You just didn't go to lectures. All right. Fair enough. Basically, these channels are used for learning. Just remember that, like, calcium within cells causes changes, as I said, and these changes in a lot of neurons can cause increased or decreased receptors, and this is how sort of your memories are formed. Yeah. So, like, don't worry about it too much. You can read over it if you want. The neuromuscular junction, I think you would have gone over this. It's quite important. Basically, the neuromuscular junction is a bit different. You have vesicles of neurotransmitter, but what they do is they cause increase in calcium within the cell, and they bind to nicotinic receptors. Probably just a word to remember. Nicotinic receptors are just very relevant everywhere. And then the action potential effectively sort of runs through the muscle, which isn't true. It's not an action potential, but like changes in ion flow and that sort of stuff happens in muscles, and this allows for muscle contraction. And then the ACH is removed from the synapse via acetylcholinesterase, and this is stuff that you'll cover in a lot of detail next sem when you do upper limb and lower limb, because that's just basically the only physiology of muscles. So yeah, that's just neuromuscular junction. Maybe worth a read through. I don't know how much your lecturer spent time on it. So yeah, just a table that you can have a look at. Just a key thing about the neuromuscular junction is, like, if you want your arm to move, like, you want it to move, yeah? Like with the brain, it's like it's very complex. It's like you have thoughts, you have like 1,000 thoughts at once, and they all just kind of add up, yeah? And you're not conscious about the thoughts that you're about to have. If I say think about an elephant, you're all thinking about an elephant. But like with a neuromuscular junction, when you want to move an arm, you want to move that arm. So you have a lot of acetylcholine, you have a lot of neurotransmitter, so that and you have a lot of receptors as well, so that like as soon as that like impulse is sent down the nerve, all that neurotransmitter is released, and it results in a lot of contraction. Yeah, and then obviously you can build upon that contraction when you're like lifting weights, that sort of stuff. But it's basically a guaranteed thing. There's no inhibitory or excitatory postsynaptic potential. It's always just excitatory. Great. Synaptic plasticity, as I said, if it wasn't mentioned, don't worry. But it's just like by using the synapse more often, that synapse is like put more on the surface, and therefore you like have memories formed. Like at the start, you didn't really know your like times table, but after doing it so many times, because those channels were used so much, now you just kind of know them. Yeah, like 5 times 6 is 30. Yeah, just like that. Thanks. Great. Spinal cord. This is very relevant in next year as well. So you have types of nerve fibers. I'm just trying to go through quickly so we can go through questions at the end as well. You have A alpha, A beta, A delta, and C fibers. Don't worry about B fibers, but I just put them in there. So A alpha is proprioception of skeletal muscle. So proprioception is knowing where things are. So when I like flex my arm, I know that I flexed it even though I can't see it. That's proprioception. A beta is mechanoreceptors of skin, which is touch. It's just normal touch. Yeah. So A alpha and A beta is like the very big fibers, the very like highly myelinated and thick fibers. Then you have B fibers and C fibers. These are very relevant for A delta and C fibers. These are very relevant for pain. And that's why a lot of pharmacology works on these, because we care about patients in pain. So the difference between them is like A delta is like sharp pain, like if you get pricked with something, you'll feel A delta pain, but C fiber is like that groaning pain, you know, like when you've got like muscle cramps or that sort of stuff. So sensory pathways to the brain, this was massive for us. Like we had one lecturer that just talked about this in semester 2. So I'm gonna just tell you it as far as I think you should know it. You have 2 pathways to the brain. You have the dorsal column lemniscal pathway, or sometimes called the DCLM pathway, and the spinothalamic pathway. So this pathway is mainly A alpha and beta nerves, and this one is A delta and C nerves. So the way I remember it is they have different pathways, and this is very important when you go next year and you'll be doing neurological examinations on patients to see if they've lost sensation in their arm or whatever. The reason like you do stuff that uses both these pathways, so that then you can be like, oh, this patient presents with an issue with this DCLM pathway, or an issue with this spinothalamic pathway. And by doing the different tests, you can figure it out. So it's quite important. And if you memorize this, it's very helpful for doing your neurological examination next year. But the way I remember it is like, think of the medulla. I'm not sure if this is true. I don't even know because I haven't done neurology yet. But think of the thalamus as the big boss, and the medulla as the boss's assistant. Yeah. So with the spinothalamic pathway, when you feel pain, you want to feel it. Yeah. So the brain's like, no, come straight to me. So you cross, because you always have to cross. You cross, and then you go straight to the medulla. Because like if someone like stabs you, you're gonna feel that. With the DCLM pathway, it's different because like, yes, you can think about where your muscles are right now, but usually on a day-to-day basis, it's not a thought going through your mind constantly. Oh, my arms are by my side. Yeah, it's not something that you're constantly thinking of. So therefore, this one goes up to the medulla, because the medulla is like the assistant, and the assistant's like, oh, does the brain need to know this right now? So it goes up to the medulla, and that's where it crosses over, and then it goes up to the thalamus. That's how I remember it. Also, the way I remember it is this dorsal column lemniscal pathway sounds really complicated, and therefore it is complicated. Like it has a more complicated pathway, and it has the more complicated sensation. Great. Hopefully no questions. If you do, just message me or whatever. Organizations of nerves, you have the CNS, don't worry about that. Then you have 12 cranial nerves that come from the brain, also don't worry about that. Basically what you need to know almost for next sem is that you have 31 pairs of spinal nerves. So you have 8 cervical, which is like in your neck sort of region, and this is what you're going to learn a lot about when you do your brachial plexus in your arm, because it's these nerves that innervate your arm. Then you have 12 thoracic, the thorax area around your rib cage, whatever. Then lumbar down here, 5 there. Sorry, the no plexus formed is supposed to be by thoracic. That's a mistake by me. Then you have 5 sacral and 1 coccygeal. So the way I remember it is you have breakfast at 8, you have lunch at 12, and then you have dinner at 5. But also 5 is way too early to have dinner, so you'll be hungry again in an hour. So you have dinner again at 6. That's how I remember it. Great. So organization of nerves. I'm just going to leave this here because this is something that you should cover have you? Yes.
What is the guys do
Oh, really? We still haven't done cranial nerves. So, as in, like, it's not a learning objective for year 1 until year. So I haven't covered it. I can send you. I can post some stuff up for cranial nerves. I'll find some stuff from third years or something, and I can post that up for you. But I highly doubt you'll be examined on it because you don't need to know it until next year. Great. So basically, just contrasting the different nerves. The somatic nervous system is very simple. You just have a cell body. It goes to the skeletal muscle and it releases acetylcholine. Now, the autonomic nervous system is different because they always require 2 nerves. Yeah. So there's always they're sometimes called the pre-ganglionic and post-ganglionic. Yeah, because you have a ganglion. That means a collection of cell bodies. So with parasympathetic, the ganglions are usually on the muscles, and with the sympathetic, you have a series of ganglions on either side of your vertebra, which you'll find out about next year when you do dissections. You actually see them; they're very cool. So they obviously do functions, and they cause muscular or like they change muscles around the body that you're not really controlling. Cool. I'll just leave that there. You can go over it. White matter means myelinated. Gray matter means unmyelinated. Efferent. This is very important because from now on you'll almost only be talking about afferent and efferent nerves. So it's important to know what they are. Afferent are sensory neurons, and I remember them by they arrive to your body, and efferent are motor neurons, and they exit because they cause the movement. And the way I remember it is like, say you were doing push-ups or basically any muscular movement, you would do them on the front of your body, yeah? Like you barely ever train out your back or do like behind backward curls or whatever. So therefore your muscular nerves, your efferent nerves come out from the ventral side, the front, and your afferent, just think tingles down your back. So the sensory ones come out from the back. That's how I remember it. Any questions so far? This is a very important diagram, and I'm going to go through it as quickly as possible in a bit of detail. So basically, this is your spinal cord. Don't worry about it too much. But as I said, tingles down your back and muscular movements in front of you. Therefore, the ventral root is muscular fibers, and your like the sensation is through the dorsal root. And also, your dorsal root needs to have a collection of cell bodies outside the spinal cord, so you have a ganglion, and it's sometimes called the dorsal root ganglion. Now, the reason why they come out from different areas is because, as I said, they're very different things, yeah? They do different functions, and therefore they come from different areas of the spinal cord. What they do is that they mix. And once they mix, that's when we call it the spinal nerve. So if you talk about the T7 spinal nerve, you're talking about it at the point where it's mixed, and it's therefore got both muscular and sensory fibers. So any nerves in your hand are usually mixed, because right as they exit your spinal cord, they've already mixed, and from that point onwards, they will always remain mixed. What they also do is after the spinal nerve, they branch into the dorsal ramus and the ventral ramus. As I said, the ramus is a collection of nerves, of different types of nerves. So the dorsal ramus is just for your back muscles and sensation just along your back, and therefore they're very, very small. If this diagram was to scale, this would be extremely thin, and this would be a very thick boy. So that's just the way I remember it as well. This is the way I used to remember it when I just started. Ventral, think front, like NT, and therefore ventral is at the front. Dorsal, because the door is at the back. That's how I used to remember it, which is dumb because the door is also at the front. But like ventral, NT for front. Also, you will learn anterior, posterior, and NT front. So it works. Cool. Any questions with this diagram, because it's quite important? Hopefully you understand it. Fantastic.
Yeah.
So yes.
Do you know what the that Weinberg Law is?
No, you'll learn about that next year. Yeah. But for example, if you had a patient with a laceration here, he would lose sensation everywhere, but he'd still have motor function. Or he would lose sensation all along the T8 spinal cord or spinal nerve area. Yeah. But if you had a patient with a laceration here, then he would lose sensation and motor function all across the T8 spinal nerve area. If you had a patient with a laceration here, they'd lose sensation and muscle movement along the front, but their back area would still have the sensation. So I'm just saying how knowing this diagram and knowing what it means can really help you with giving a clinical diagnosis next semester when you start doing these neuro examinations. Yes? So that's a good question. So a ramus is, it's still like, it's a collection of those axons together, and a plexus is when, so the ramus will then divide into specific nerves, Like you might have heard your radial nerve, your ulnar nerve, medial nerve. Maybe you haven't. Maybe you'll learn them next semester. But like what a plexus is it's when these nerves come out and they start mingling. But that was a good question. Does that make sense? Sort of, yeah. I mean, it'll make more sense when you do it next semester. It's kind of hard to explain, but a ramus is when it just exits out of the spinal nerve, and then it divides, and then it reconnects back, and that's a plexus. Cool. Sympathetic trunk, I kind of talked about this. Don't worry about it too much. It's there if you want it. Other miscellaneous things. Interneurons, important for the reflex arc. These are just notes that you guys can read, but you have an inhibitory neuron and a normal interneuron. So basically, for example, with the knee reflex, yeah, you have one muscle that's contracting, that's a quadriceps contracts, and obviously the other muscle, your biceps femoris, needs to relax. So that's why you have an inhibitory neuron, and that sort of just loops back. So you have that sensation coming, and it loops back, and it causes that reflex. Great. Gate theory of pain. This is very important. It'll come up a lot. But basically, you have your C fibers, which are causing the pain, and you have your A beta and A alpha fibers as well, and that's like proprioception, mechanoreception. And what your brain does is it thinks these fibers are more important, and therefore what happens is if you really hurt your arm, that's why massaging it really activates those A alpha and A beta fibers, and it like overpowers that pain response with that proprioception and mechanoreceptor response. So it's really effective. That's why massage works, because the brain just thinks that the pain is less important, which is kind of dumb, but it really works therapeutically. So referred pain, this is when two sensory things enter at the same area. For example, someone presenting with a heart attack might have arm pain, and that's because the nerves which innervate the sensation of the heart enter at the same level as the nerves which innervate the sensation of the arm. And your brain's just not used to your heart telling you that it's got pain. So your brain will just get confused and be like, oh, this must be my arm that's feeling the pain. And the same thing happens with a lot of conditions, because patients will never present with like, oh, my duodenum is in pain. They'll just say, I've got pain on my like stomach area. Great. Enkephalin is the body's own opioid, and therefore you have receptors for it. It just causes hyperpolarization, and therefore the pain sensation, the pain signals can't be sent along. Great. Any questions for me before I go through a few questions quickly? No worries. I will answer any questions later, or just message you. Feel free to message me. I have 10 questions. I'll go through them. If you don't want to stay for this, that's fair enough. But the way we'll do it is I'll give everyone a second. So don't call out the answers for a few seconds, and then once you think you know the answer, put your hand up. First person to put their hand up gets a chocolate. Cool. All right. Quran's disqualified. No, I'm joking. You can. Cool. Answer the question. Muscle and why? Yep. Cool. Muscle, because also the neural tube is just ectoderm. Great. Only motor neurons will be found where? Which of these will you find only motor neurons? You know the answer one question. But you can put your hand up as a flex. You put your hand down. So yes. Ventral horn. So the horns are parts of the spinal cord. Yeah, ventral root. There you go. Great. Hopefully that makes sense. Which of the following is not a function of the sympathetic nervous system? I didn't really cover this, but I did. You were first. I'll give everyone a second. Wait, do you know it, or did you just put your hand up? Okay. Yes. D. No. So the sympathetic nervous system does both contraction and dilation, and that's one of the key things about the sympathetic is that the parasympathetic does not innervate any blood vessels anywhere except the penis and the clitoris, and it's contraction of the bronchi, because the bronchi are the passageways for the lungs, so constricting them would mean less air, which is less ideal. At which stage of the action potential is the voltage the most negative? You were first. Hyperpolarization, great, and I'll just pass this halfway. Sorry, yeah, someone? That's all good. Cool, hyperpolarization. Easy. Any questions so far? Great. Which of the following is false? There are a high number of voltage-gated sodium channels at the axon hillock and the nodes of Ranvier. Voltage-gated potassium channels open during repolarization, but not depolarization. Chlorine channels are used to stimulate an excitatory postsynaptic potential. Calcium channels are essential for neurotransmitter release. Sorry, I wasn't really looking at who put their hand up first. So just whoever puts their hand down first. All right, that was you. Cool. C. Yeah, cool. Because the calcium channels are negative, and therefore they're more outside. So opening the chloride channels, the chloride will come inside, and they would cause a more negative membrane potential. Which of the following only contains inhibitory ion channels? This is a bit of a hard one, but you can extrapolate. D. Yes, correct. I don't know who said that, so I'll just throw this out there somewhere. Nice. Which of the following is false? Yes. B. Yes. Where are you? Oh yeah. Sorry. Cool. Which of the following is false? This is a hard one, to be fair, but you'll be right. It's false. Which of the following is false? Yeah. Yes. C. No. So the DCML decassette at the medulla, if you remember, because the medulla is like the secretary or whatever. So it's not vibration. But at this point, that sort of question is unreasonable, but this is stuff that you'll be expected to know soon. I'll just throw one out there as well. In the gate theory of pain, when stimulated, C fibers directly stimulate the projection neuron causing pain. And the others, I can't bother reading it. I'll give everyone a second just to read it. Yes. C. So no, because B fibers. Anyone else? Anyone else? There's a hard one too. Yes. D. Oh no, it's B. So it would not cause pain. It would cause no pain, and muscle movement is the A alpha fibers. So that was a hard one. It was just kind of putting two things together. But yeah. All right. Thanks for coming. If you have any questions or find anything that's wrong, please message me and I'll fix it. Yeah. Cool. Karan. Thank you. Oh, okay. Yeah, yeah. That's really good stuff.